Welcome to the next video in our training series. Now that you have a bit of project background, we're going to move on to the protocol for collecting seed and data and share some collection tips. One of our goals with this project is to train volunteers how to properly and sustainably conduct a seed collection, and we hope the skills you learn through Project Wingspan will transfer and enable you to contribute to future conservation projects in your community. All information provided in this webinar is also available for review in our seed collection manual that may be downloaded directly from our website. To become a seed collection volunteer, you will need to complete the following steps. First, watch each of the 3 online training videos. Next, fill out the contact information sheet on the website and send a completed copy of the volunteer waiver form to James Hart via the email address you see on the screen. Once this is complete, you'll be connected with a local collection team and will be notified about seed collection activities in your area. Then you'll be ready to get out and start collecting seeds. Seeds and the vessels that contain them often come in different shapes and sizes. On the screen here are just a few examples from the species collected through this effort. From left to right, you can see the relatively large and bean-like lupin pods, the small, round capsules of blue-eyed grass, a slender California poppy pod that bursts open to disperse seeds, a dry, open milkweed pod releasing its wind-dispersed seeds, and the light, fluffy seed heads found on a goldenrod. With this diversity of seed morphology and dispersal methods, it's important to understand how to identify and properly collect these species. Through this training, in the materials found on our website, and through getting out into the field, you'll learn how to do just that. Pollinator Partnership staff and partners will scout the sites and see if the seeds are ripe or ready for collection. However, it is still important for volunteers to know the difference between a mature and immature seed or seed pod. Not all plants will mature at the same rate, and while there may be enough mature seed to host a collection of a plant population, there may still be unripe seed mixed in. Generally, you'll want to look for changes in the fruit and seed color. Ripe seeds will be darker than unripe seeds, usually turning from a paler green or white color to brown, tan, or black. If the seeds are housed in a capsule or pod, the capsule will dry out and turn a brown, grayish, or tan color when the seed is ripe, as you can see in the blue-eyed grass and lupin to the right. If you shake the pods, you can often hear a rattling noise as the seeds are dry and loose. Also for seed found in capsules, the fruit will split or break open when the seeds are ready. Look for a crease or ridge that appears just about to open or is already slightly opened. This can be an ideal time for collecting, as the seed is ripe but has not yet been dispersed. That said, don't worry if some seed has dispersed already. It is an inevitable part of seed collecting. Finally, when you have seeds in your hands, they should be plump, dry, and hard. Timing of collections is important, as you want to capture the seed in the sweet spot where it has ripened fully, but before it is entirely dispersed. This is particularly important for wind-dispersed species like milkweeds. The pod on the left here is not ripe yet. If you were to collect at this point, the seed would not be viable. However, as you can see on the right, if you wait too long, the pod will open and the seeds will be carried off in the wind, and you'll lose out on a chance to collect from the plant. Ideally, you'll collect from these pods when the pods are just starting to split open, showing that the seeds are ready but still mostly enclosed and safe from the wind. When this is the case, you can open the milkweed pod and scoop out the seeds. One way to test whether or not a seed is ready is to perform a seed cut test. A cut test is a way to inspect the internal tissues of the seed to check for maturity and viability. If you cut the seed open and the inside is firm with a fully developed embryo, then that seed is mature. Similar to a cut test, you can also perform a squeeze test. Squeeze the seed, and if it is firm with a dense outer coat, then it is likely mature. If you perform the cut test and more than 30% of the seed that you test are either empty or bug infested, it'd be best to move on to a different population, or collect more seeds to compensate for the non-viable ones. A cut test will not work for every kind of seed, because some are simply too small, so you will have to rely on visual assessment for those species. In order to reduce the likelihood of contamination of our seeds by insects, fungus, or other factors, it is important to only collect the seed heads, seed pod, or seed whenever possible. Do not collect any excess materials, such as stems, leaves, or rocks. If stems or leaves are collected, we ask that you please remove them before shipping the materials. Please keep an eye out for insects as well, as they can pretty easily hitchhike on these materials. Additionally, never put anything except plant materials in your collection bag, to prevent any items from getting lost or broken. If a collection is found to be contaminated with weed seeds, it will be discarded to ensure that this program does not accidentally spread any invasive or otherwise unwanted species, such as those on screen. We don't want all of the hard work that goes into scouting, planning, collecting, and cleaning seed to go to waste. To prevent accidental weed seed contamination during the collection, make sure to walk with your collection bag closed. And if you are collecting with both hands and setting the collection bag on the ground, just make sure there is nothing weedy above it that may disperse seeds into your collection. Lastly, be mindful of your clothing and make sure that if any weed seeds latch onto your clothes or your sleeves, that you brush them off before putting your arms into the collections. Now, let's talk about seed collecting techniques. You will want to collect randomly from a diverse selection of plants, regardless of their characteristics. We want to make sure that we are getting genetically diverse seeds that will be suitable for reintroductions. Even if an area has very dense plants, spread out and collect from other individuals throughout the landscape as well. Try to include all of the microhabitats throughout the site, such as areas that are wetter, drier, or on a steep slope. Incorporating those plants into the collection will help increase the genetic variation. Make sure you are not collecting only from plants that look the biggest or the most showy. You don't necessarily want to avoid short, scraggly, or otherwise different looking plants because they may have important genetic characteristics that could help them flourish under different conditions. Techniques will vary depending on the plant, and your collection lead can advise you on the best techniques once you arrive at the site. We want to make sure that when collecting seeds, we don't harm the collection site populations, and there is still plenty of seed left to perform its natural ecosystem functions. Best management practices, according to the Bureau of Land Management Seeds of Success program, is to collect no more than 20% of any individual plant's reproductive output in the year. You can do this by collecting from every fifth plant, or collecting 20% from each plant you pass. You will want to leave around 80% of the seed at the site to ensure there's plenty to replenish future populations. As I mentioned earlier, there are many different types of seed heads out there, and that's true of our target species list as well. Several species targeted through this effort have seeds housed in capsules. California poppy has a long, slender pod that splits open lengthwise when ripe, throwing seeds a foot or more away. This pod should be collected before it opens, but after it has dried sufficiently and looks a tannish brown color. You can remove the entire seed pod with pruners and place it in your collection bag. Milkweeds were described earlier, but they should also be collected when the pod is dry and splitting open, but before the seam on its side opens too wide. Collect the seed by removing the pod with pruners, splitting open the pod more with your hands if necessary, and scooping the seeds out with your fingers. You will likely get quite a bit of fluff with the seed, and that's okay, although you should be careful to keep the seeds in the fluff out of the wind as they can get swept away easily. Some plants, like blue-eyed grass and clarkias, have capsules that dry and open at the top when the seed is ripe. It's best to collect these species just before or just as the capsule opens. For blue-eyed grass, it's pretty easy. Simply remove the dark brown or black, ripe capsules with pruners and place them in your collection bag. With clarkias, remove the entire stem, which may contain numerous seed capsules, and shake it upside down into your bag. This will release the ripe seeds into your container. One other thing to keep in mind is that seed should only be collected when it is dry. Do not collect seed when there is morning dew present or after a rain has fallen. If you do, it can bring excess moisture that can lead to mold growth and spoil the seed. Many plants in the Asteraceae family, such as woolly sunflower, yarrow, and gum plant, develop a relatively flat, round seed head that holds the seed upwards. You can often see the seed from above, and if you rub the seed head in your fingers, you will dislodge some of the seeds. For these species, it's best to remove the entire seed head and place it into your collection bag. Some plants, such as goldenrod, milkweeds, or Pacific aster shown in these photos, use the wind to disperse their seeds. Species that use this method have papas or scales, bristles, or feather-like hairs that attach to the seed and help it carry in the wind when it's ripe. This fluff acts as a parachute, and, if you're not careful, your seeds will fly away and evade collection. When collecting from species that utilize wind dispersal, it's easiest to cut off the entire seed head from the stem and place it in your bag. Then, once you're in a sheltered area away from the wind, you can separate the seeds and fluff from the stem. You do not need to worry about removing the seed from the fluff, but make sure to remove any stems, leaves, or stalks before shipping. An ideal collection would result in at least 1 ounce of clean seed. Collections resulting in more than an ounce are always encouraged whenever possible, as the more seed we can get, the more habitat we can enhance. For reference on how much seed is in an ounce, you can see 1 ounce of 3 different species here pictured in a plastic water cup. In each case, the seed takes up about the bottom third, or roughly 1.5 inches, of the cup. It's important to note that there can be a big range in the number of seeds needed to get to this weight, as yarrow can have well over 100,000 tiny seeds in 1 ounce, while something like milkweed or garden sunflower may only have a few thousand seeds. Data collection is required for each seed collection activity. It is required that you fill out our online seed collection form that can be accessed through the link provided by your state coordinator or team lead. The first section of this page should look like this on the right. This section of the form is information about the seed collection event and those who are present. Once this is complete, you will see a map further down the page. There are a few different ways to find your site on the map. If you are at your collection site location, the easiest way is to enable location tracking on your mobile device and use the location setting on the map. If you are located near your site, you can find it by scrolling through the map to locate the site from your current location. If you are not located near your site, enter the address or nearest address of the site in the search bar, and the site will be brought up on the map. Once the site is located, use the polygon tool in the upper right corner to locate the borders of the site as accurately as you can. If you have any questions about this process, there's a step-by-step tutorial on the Project Wingspan webpage. Seed should not be collected when it's wet, but regardless of whether or not your seed appears dry, it will require a 3-day drying period to get rid of any residual plant moisture and allow any bugs to scurry away. Drying and cleaning seed will look different for every team depending on the facilities available. Your collection lead will let you know if they need any assistance cleaning seed before shipping it off. Collections should not be kept for long periods of time before shipping to ensure the seed maintains maximum viability. It is okay to hold on to collections for a week or so if they're stored in a dry, dark, cool location with proper ventilation in order to ship multiple collections in bulk when possible. Collection leads are responsible for the shipping of the seed, and all collections will be sent directly to heritage growers for cleaning and testing. Pollinator Partnership staff will provide the information required to print a UPS label and ship the seed. There are some safety concerns to consider when participating in a seed collection event. Milkweed has thick, white sap that can be irritating to the skin and eyes. Consider wearing gloves when collecting and be mindful not to touch your face until you've washed your hands. If milkweed sap does get into your eyes, rinse thoroughly and seek medical attention. Seed collections can take place throughout the year, and conditions for collecting can range from hot and humid to cold and windy, especially in California's various microclimates. Make sure you are staying hydrated, wear sunscreen, and dress for the weather. Also, always dress in bright clothes if collecting near a road or as required by site specifications. Ticks are found throughout our collection region, so make sure to check for ticks after leaving the field. A tip we recommend to avoid ticks is tucking your pants into your socks and your shirt into your pants to prevent ticks from getting under your clothes. Lastly, make sure you're aware of your surroundings and keep an eye out for poison oak, even if there aren't any leaves present. In natural resources work, including seed collections, equipment is often moved from one location to another, leading to the potential for the spread of non-target species. Non-target species include the plants, animals, diseases, pathogens, and parasites that are not intended to be moved. To reduce our negative impact and prevent the movement of invasive or noxious species, please educate yourself on weed identification in the field and be cognizant of seeds that may cling onto your clothing and gear. Cleaning dirt and plant materials from your shoes and other materials is especially important when you're collecting in areas that may be affected by sudden oak death. Not only is this important work, but it's also a lot of fun. Please feel free to take photos of your collection teams in action and send them our way or tag us on social media. We can use these photos to further promote Project Wingspan and recognize volunteers for all of the vital work they are doing. An important factor in judging the success of a project is volunteer engagement and community support. We measure this by keeping track of various affiliations and time spent by volunteers. This is important information to us because it is reported to the National Fish and Wildlife Foundation as part of our regular grant reporting. Thank you for joining us for this module of the training series, going over seed collection protocol and collection tips. We greatly appreciate your interest in pollinator conservation and becoming a trained volunteer for Project Wingspan. We're excited to have you join our growing network of volunteers, making a tangible difference for our imperiled pollinators.